Hello, welcome friends. Welcome to Datta Biology class. So we are continuing our discussion on DNA replication. On the earlier episode, I have said what is DNA replication. So every year, whenever it comes in Michelson cell experiment or DNA replication, you first write the definition. You should first write the definition. Definition is very important. You will write that DNA replication occurs in the cell. In the cell replicates or the cell make copies of its DNA, and it occurs in the S phase of cell cycle. So why it occurs in the S phase? Synthesis phase. What is there in the synthesis phase? We have not gone through anything about the synthesis phase. Only in mitosis we have mentioned that. Why it will occur in synthesis phase? That we have to learn. Then it will be clear for us. Okay, let us. You please go to my mitosis episode. Then you will find that I have detailed discussion on this. Here I am having a little touch on that. In synthesis phase, S phase, what actually happens? Suppose the cell, this is the cell has to undergo, this is the nucleus, and within the nucleus whatever was there. Okay. Now this cell will undergo mitosis. In mitosis there are two steps, karyokinesis, cytokinesis. So karyokinesis, the nucleus will divide, and cytokinesis, cytoplasm will divide. Now, in nucleus, for division of nucleus, we mentioned prophase, metaphase, anaphase, and telophase. These are the four phases we have segregated, but all the steps goes on dynamically. So there is no boundary that this will be up to prophase, this will be up to metaphase. No, this is a dynamic process. It goes on. So what we have seen in prophase, the chromosomes becomes condensed, and in metaphase, the chromosome structures are studied because the chromosome appears clear. Now, in the metaphase, the chromosome appears like this. In chromosome, we have seen there is centromere and there are two chromatids. Isn't it? So, in metaphase, I have said in my earlier page, you go to mitosis, you will find it. Suppose this is the cell. The cell, you fill the cell with half water or cytoplasm. So it is half of cytoplasm here, and then there was two centrioles. The centriole divides one centriole here, one centriole here. They form the spindle fibers, and in metaphase, the chromosome, this chromosome comes here. This chromosome comes here having two chromatid. One chromatid in this way, one chromatid in this way. This is centromere. That we have seen in metaphase. The metaphase, the chromosome comes in the metaphase like a metaphase plate. In human, there are 23 pairs. That is 46 chromosomes lie in the, here it floats, the 46 chromosomes. And what we have seen that this fiber is known as chromosomal fiber. This fiber is known as chromosomal fiber. and this is continuous fiber. Now what actually happens? This is a chromosome. It is a part of single chromosome. Clear? Now, this chromosome in the anaphase stage, it goes to half goes to this side, half goes to that side. So I will write this in this way. The cell, say half of the chromosome will go here, and half of the chromosome will go here. The chromosome are made up of chromatids. Two chromatid forms a chromosome, and here you see the chromatid tears up, splits, and there are some fibers we used to call that interjunal fibers. Clear? And this fiber is known as chromosomal fiber. This is chromosomal fiber. So half of the chromosome is going to this side, half of the chromosome is going to that side. But we used to call this as, and thereafter this chromosome comes here. The chromosome comes this is equatorial division, this is polar region. This chromosome will come to polar region. This polar region, this chromosome will come. And the chromosome is made up of two chromatids. Here it is made up of single chromatid, mind it. And it is the chromosomal membrane will form. Okay? Chromosomal membrane will form. And now cytokinesis will take place. Cytokinesis will take place. So, what we have seen, from this chromosome to cell, from this cell, two cell has been produced and two daughter cell has been produced, but actually it is equational division. It was having 23 pair of chromosome, that is 46 chromosome, all 46 come, all 46 tears up, and 46 piece goes to this side, 46 pieces of chromatid goes to this side, they form chromosome. Why they will be called chromosome? Because in chromosome we have seen that it contains two chromatids. Whether here it is two chromatids? No. So here this 46 chromosome will have a single chromatid. Okay? We know that these chromatids are made up of DNA. And after this cytokinesis, the cell will take rest. That is known as interphase. Interphase is divided in gap 1, gap 2, and this is known as synthesis phase. Clear? In this synthesis phase, after this daughter cell has been produced, they are actually daughter. They are not able to reproduce or go for another division. They actually they take rest in the gap 1 and they synthesize their DNA, because this chromatid are made up of single chromatid. So in synthesis phase, they will produce another chromatid here and they will form the actual chromosome. Actual chromosome is there. And after forming this, they will take a gap of another little bit time and they are ready for further cell division. So in the synthesis phase, actually, what have been told by us that two daughter cells are produced and they are having the same chromosome number. Although they are having the same chromosome number, but their chromatids were single. But they synthesize their chromatids, that is, they replicated their DNA. So DNA replication occurs in synthesis phase and it occurs after the cell division is over. In the synthesis phase of every cell division, the DNA synthesis takes place. So DNA synthesis occurs in cell cycle. In the cell cycle, the cell make copies of its DNA. Okay, and transmits the cell cycle and transmit their heredity material. So what we say definition? Definition that is replication of DNA is a process of which occurs within the cell, and within the cell, in the cell cycle, the cell make copies, two identical copies of its DNA and transforms their heredity material. Okay. It occurs in synthesis phase. Now we will start with the main process of DNA replication. Still we have not entered. Okay. Now we see what happens. Where is the DNA located? In the cell, we have nucleus. In the nucleus, we have chromosome. Chromosome remain covered by membrane. These are proteinaceous membrane, mainly of histone protein or chromatin protein you used to say. And then these DNA in synthesis phase, this is DNA, this is DNA. This DNA are replicated or make copies. Now, anybody cannot come and change the DNA because it is so precious. DNA is so precious that if anybody come in a fine morning and change it, suppose you are working in an office where the entire office goes on through a computer LAN system processor like that, and there are servers are there inside the office. So whether anybody can go and change the structure of DNA or applications of DNA, no. They should get security checked first. So DNA replication, before DNA replication, the DNA machinery should be allowed to enter the chromosome, to enter into the chromosome to make copies of this, because they will get access. So in the first phase, what happens? In the first phase, some access is required. Okay? That access, how it is accessed? Because the cell nucleus within itself gives the chromatin protein instructions that we are going to replicate our DNA, so you loosens your protein. So chromatin protein loses. Chromatin protein loosens to allow access to the replication machinery. They allow access. Otherwise, nobody can enter and change it. So they are losing its security. That is, these securities are standing one after another. Nobody can enter. Though there is an instruction, yes, we loosen it. We loosen it. So there is an security structure on the instruction of the nucleus that this will be replicated. Okay, this machinery consists of replication enzymes is required, replication enzymes is required, and RNA is also required. And for doing this work, some energy is required. Because without energy, how the labor will do this work? So replication, to enter the replication machinery, they first allowing access to this. First of all, what they will do, we will go for the energy. They will go for the energy. What actually happens in the nucleoplasm? In the nucleoplasm, there are, this is nucleoplasm, there are adenosine monophosphate. In the nucleoplasm, guanine monophosphate, thymine monophosphate, AGTC, cytidine monophosphate. These are available in the cytoplasm. So this had to be activated to do the work, because ATP, we know the NDP is energy, GTP is energy, TTP is energy, CTP is energy. So first we will do, you write your definition, and then first security check, definition, then you will write security check. That is allowing access to this. Number 3, you will write transformation, or you can say activation. Activation is activation of, these are called deoxymonophosphate, deoxyribonucleomonophosphate. These are actually called adenosine monophosphate. No, adenosine is the particular name, proper noun. These are called deoxyribonucleotide monophosphate. So activation of deoxyribonucleotide monophosphate to deoxyribonucleotide triose phosphate. Triose phosphate for supply of energy. So whatever AMP, GMP, TMP, CMP are there in the nucleus or in the nucleoplasm, they get converted to deoxyribonucleotide triose phosphate. AMP, what will be there? ATP will be produced. So from AMP, ATP will be produced. Likewise, GTP will be produced, TTP will be produced, and CTP will be produced. That is, the replication machinery is now ready with energy. So power supply has been maintained. So there is a full power supply for replication of DNA. Okay, first you will write that number 1 definition, then there will be the cell will allow replication machinery free access to the DNA for replication. Okay, and for that case, the chromatin protein, mainly made up of histone protein, they will loosen themselves to allow the machinery, replication machinery to do the replication of DNA. Then first activation of deoxyribonucleotide monophosphate to triose phosphate. By this way, AMP, GMP are other. This has been done. You can write one line more, that this is known as phosphate is adenosine monophosphate is, this is adenosine I told you it remains joined with sugar. It remains joined with sugar here, CH2, OH. This is C1 position. So adenosine, this phosphate is there. So it is known as adenosine monophosphate. But in true sense, it is known as deoxyribonucleotide monophosphate. But it is proper noun is adenosine monophosphate. Then another phosphate is added. Then another phosphate is added. So it becomes ATP. This is done by, this is known as phosphorylation. This is known as by ATP, AMP, GMP, TMP, CMP will be converted to ATP, GTP, TTP, CTP by a process known as phosphorylation. You write phosphorylation takes place with the help of enzyme like phosphorylase. Phosphorylase. This will be the first one. We'll go into the detail to take a clear view of the process of this activation. So it is the activation of this. Okay, erase this one. You pause and change, write it accordingly. Now we have already produced this, and we have produced these bases. Nucleic acid bases we have produced in triose phosphate position. Suppose we have produced adenosine, sugar, and phosphate. One base we have produced. Likewise, 2, 3, 4 bases. 4 bases we have produced. Although they in the nucleotide they are having one single P phosphate, but we are ready with another P. Whenever this bond will break now, it will release energy. Anyway, now these two things we have prepared. Before replication, what we have seen, the DNA is made up of nucleotide A, T, G, C, T, A, C, G. These are joined with double hydrogen bond or triple hydrogen bond, as the case may be. This is a purine and pyrimidine basis that we have come across. Now here we have seen, here one sugar is there, phosphate is there, sugar is phosphate, sugar, phosphate, sugar, phosphate. This is nucleotide, deoxymononucleotide, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. So what we have to see that all together we have got 4 bases, 1, 2, 3, 4. These are all same. So the restriction of this pairing of purine and pyrimidine bases, the restriction of pairing is there, and all together we have got 4 bases. Now the next, before replication, what we will do, this has to be unchecked, or this has to be separated. So we write the next point after activation. Say it is 1. Now this is 1. Okay, make it 1, make it 2. After activation, unwinding, or you can write unwinding, unwinding of DNA. This will be my job. Replication machinery's job will do unwind this. They will take some machinery, some carpenter, some everything, you will go. They will take helicase. They are the carpenter. So helicase will come, helicase will cut here. So what we have to do, we have to unwind this. Unwind this. oh, sorry, this is not been unwinded right now. Till now it is like this. We have to unwind this, cutting these pieces here. So if we unwind this, that somebody says that it starts from 2 hydrogen bomb because it is, weaker as compared to this. Okay, no matter. So here some enzyme will come. That enzyme will come to unjig it. That enzyme will write with the help of some enzyme called this is a scissor. Scissors. This is known as helicase enzyme. So replication machinery contains replication enzymes. Their enzyme is known as helicase scissors. So scissors will come here, it will cut, it will cut, it will cut, it will cut. So they will be cutting this, and as a result, it will unjig. This will tear open. So this is tearing open. It is tearing open this. Here one thing I like to mention, that in bacteria the DNA is circular. So if it starts circular like this, say for example, if we start from here, so it will go on like this. So there is single initiation point or replication point. But here there may be so many of these. How? This will be inflated like this. This will go inflated like this. This will go inflated like this. So there may be several replication, producing several bubble. These are called bubble, replication bubble. Not yet formed. These are called replication bubble. And here it will cut the hydrogen bond. It will cut the hydrogen bond. So cutting the hydrogen bond will not be sufficient because two things are there. First things that the AGTC are here. A, T, G, C, A, T, C, G, A, T, T, A. In this way, it were there. ATGC, in this formula, they were binded like this. In sugar and phosphate also. So actually, it is like this. I am writing it here. Adenine, sugar, phosphate. Here, thymine, sugar, phosphate. And then guanine, sugar, phosphate. C, sugar, phosphate. T, A, G, T, C. Two will be enough. Another one I am writing. T, sugar, phosphate, A, sugar, phosphate. So, whenever I have unwounded this one, this is like this, and this is like this. It remains joined this. Okay. So, it was there, the hydrogen bond were there, 82 hydrogen bond here, 3 hydrogen bond here, 2 hydrogen bond. We have cut this. And these are all joined up with glycosidic bond and phosphodiester bond, like that. So, if we cut this now, after some time we will see that they will make new bond. they will make new bond. They will make new bond. So my purpose will not be served because I have entered to replicate it. So in order to check this, so that it does not come closer, we put some enzymes here. These are contained in replication machinery. Replication machinery enzymes are we will put. These are called single strand binding protein. Clear? So these are called single strand binding protein. So we are fixing. Suppose this is a nail, we are fixing it. We are fixing it in this way so that it cannot come closer. So we have teared it up. We are unzipping or unwinding the tail to protect the realigning or so that the two strands doesn't come together. We have put the single strand binding protein. Okay. There is another problem because this remains in a helical state. In a helical way, because we told that it is in helix. It is in helix. So some helix stabilizing enzyme should be incorporated here, because here I will incorporate the helix destabilizing enzyme. Is the common name. Helix destabilizing enzyme. These are known as topo isomerase. With the help of helix destabilizing enzyme, what we are doing? We are not allowing this to recoil again. So we are putting some enzyme, helix destabilizing enzymes known as topoisomerase in human or eukaryotes. Some bodies are saying that gyrase, G-Y-R-A-A-C, gyrase. So here is gyrase, G-Y-R-A-A-C, gyrase or topoisomerase. You can write here topoisomerase. So unwinding is almost successful. Whenever we are tearing it up, as soon as it is written in so many books, everybody is saying as soon as we loosen your pressure, so it recoils again. Write that also. And otherwise you can write this to make the stable two strand. For destabilizing, we are using helix destabilizing enzyme. In the replication machinery, we have gyrase, topoisomerase. These are helix stabilizing enzyme, and we are using SSB. SSB is included in replication machinery. These SSBs are there to fix. Okay. Now, the two strands should separate before, because these two strands should separate before it serves as a template. How? What is template? Template is nothing. Template means you are making new copies of this. Two identical copies. It is going actually here, you see, some new strand will be formed. The new strand will have this. New strand will have this. On the other hand, here you will make a new strand. Here it will have A. It will have C. It will have T. And in this way, it will have G, T, G, and A. So, one is complementary to other. So some complementary strand should be prepared. In this way, in Michel's and Shell's experiment we have seen the proper complementary strand should appear. Now, we are prepared with what? For replication. Okay. Here we will have, say, we will write simple way A, T, G, C, A, T, G, C. The reverse is there. A was binded with T, T, A, G, C, D, A, T, T, A, G, C, C, G. In this was, it was there for unzipping. Now, there is the formation of replication bubble. This replication bubble, and we have 4 templates. We have 4 templates ready. A, S, P, P, P. This is one. It is ready. Adenosine, sugar, phosphate, phosphate, phosphate. Guanosine, sugar, phosphate, phosphate, phosphate. AGT. Sugar, phosphate, P, P, P. AGTC, C, sugar, phosphate, P, P. Sorry, 1 P, P, 1. There will be 3 P. So these 4 bases are ready. We are ready with that. We are ready with the energy. Energy also there. Even if we are ready, we cannot prepare it. And I have not mentioned, this is 5 prime to 3 prime, and say this is 3 prime to 5 prime. That we have discussed. Now, another enzyme will come. This is, now here some primer will come and binding with the new bases. So, in next step, you write this I am erasing. Number 3, you write primer and binding. So we have got binding blocks, but still we are not having primer. Why primer is required? Why? Because DNA, another replication machinery, contains DNA polymerase. I will come, there are 5 types of DNA polymerase. I am coming. So DNA polymerase will act from 5 prime to 3 prime. It starts from 5 to 3. So 5 to 3 fits here. The new strand will be here like this. This will be 5 strand. That is 5 3. It was 5 3 all 5. This will be 3. This will go in this way. So here you will see that here TAC is there. DNA polymerase cannot fix this because T will have A. So this will come. A will come here to fix. Is it not? A, T will come here. In case of A, this T will come here. In case of C, G will come here. So they will take, DNA polymerase will come and fix here. But they do not know where from it they will start. So it is the RNA primer. Actually, it is known as primer. DNA polymerase, I have written primer. This is actually RNA primer. Primer, these are actually formed from DNA polymerase. This primer we will make a short primer. This will make a short primer here. And it will give intimation to DNA polymerase. You start from here. So this is the RNA primer, or in short it is known as primer requirement or primer. So some primer will come from the replication machinery and they will fit it. And what will be done? DNA polymerase with smell will sense that here is steam. So DNA polymerase will bring the adenosine or deoxyadenylic monophosphate, triose phosphate here. So this triose phosphate will come here, not monophosphate. Mind it. Here in earlier cases, whatever DNA was there, it had only one P. Now here I am bringing, it has 3 P. Is it not? So it will come, it is known as elongation. So whenever it will come, what we will do, in T, 2 hydrogen bond, then ASPPP will come. Is it not? A, sugar, phosphate, another phosphate will come. It will sit here. And thus of A, T, S, P, P, P will come. Here, T, S, P, P, P will come. It will join with T, A. It is A here. And C, in case of C, suppose it is C, G will come here. G, S, P, P, P. G, sugar, phosphate will come here and join. In this way, they will go on joining like this, because the primer has instructed them to start their strand like this way. Will come what is leading strand and lagging strand. And now let us complete this first, will come later on that one. Here we will see, after fixing with this, they will give up this P and P. They will give up in the nucleoplasm, they will give PP. They will give PP. They will give up their PPP. That is known as deoxyribonucleotide triose phosphate will get rid of their PP, that is pyrophosphate. This pyrophosphate will be released in the nucleus itself, within the nucleus environment, and this pyrophosphate on hydrolysis, you will write, on hydrolysis will produce inorganic phosphate plus energy. This energy is responsible to fix this. Where from energy will come? This pyrophosphate on hydrolysis will produce inorganic phosphate and will release energy. Will release energy. That energy is required for DNA replication. Now, on the next episode, we will start the leading strand and that strand. And thereafter, a little bit here, we will complete that on that. Till then, I am taking a break today. Thank you very much.